In earlier class, we have seen a disease which is caused by a virus, that is hepatitis A type of virus, which causes hepatitis A type of infection. And this infection, it only occurs in case of human beings. And this hepatitis A type of infection, we have seen, it is mostly affecting the human beings, but it is mostly affecting the human beings, and it is highly infectious. Means it can spread very rapidly among the healthy persons because of the that source. And that source which we have discussed in earlier type of hepatitis A, the source of infection is the contaminated water or food, and through which this infection can be initiated in case of healthy persons. Now, the second type of infection that we are going to see today, that is a hepatitis B type of infection. And this infection is also known by another name, transfusion hepatitis, serum hepatitis. And this is called because this type of hepatitis is mainly transferred through the blood transfusion process. If that blood is contaminated with hepatitis B type of virus, then it can be easily transferred through this blood into a healthy person. And because of such type of transfer which occur because of the using serum or blood, because of this it is known as transfusion type of hepatitis. Now this hepatitis B type of virus, it is most widely spread and most important type of viral hepatitis. And in Marathi we call this infection as a jaundice. This infection, it is known as a jaundice, and it is one of the most important viral infection which occurs in human beings. And when it is estimated that more than a third of world population have been infected with hepatitis B type of virus. Means whatever the population we have today of that world, out of that whole population of the world's, a third of world population, it is infected by hepatitis B type of virus. And of all these infected, a quarter of them become hepatitis virus carriers. So, because of these facts, this is one of the most important viral infection which occurs in human beings. Now, in that, firstly, we see the etiology of this hepatitis B virus. This hepatitis B virus, it is having size 42 nanometer, and this virus is a particularly a DNA carrying virus. Means its genetic material, it is a DNA. And this morphology of this hepatitis B virus, it shows that it is composed of two envelopes, the outer envelope and the inner envelope or inner core, which is having size of 27 nanometer in diameter. And these two envelopes, they are having inside this, there is a presence of viral genome, that is the DNA material. And along with this DNA material, there is a presence of an enzyme which is particularly observed in this virus, is the DNA polymerase enzyme. And because of these unique features which are shown by this hepatitis B virus, these are arranged in a separate family. In case of, while we classify this virus, these are contained in a separate family, and that family is known as a Hepanodoviridae. This Hepanodoviridae family, it contains two genera. First genera is orthohepanovirus, which mostly contains this hepatitis B virus, while the second genera it is a AV hepanovirus. This mostly contains Peking duck hepatitis virus. So this virus belongs to a family, and that family which is known as a Hepanodoviridae family. Now this virus belongs to a Hepanodvirus type 1, and this is a DNA having virus. Means its genetic material, it is composed of DNA. Now along with these characters, some other characters are also observed there. In 1965, Bloomberg, while studying the serum lipoprotein in case of human beings, they observed that in case of Australian aborigine, this is a one type of category of human being which is present in Australia. In this type of category of peoples, there is a presence of a new antigen in that person's serum, and this antigen, he called that antigen as Australia antigen, and this antigen, it is participated in the precipitation reaction when two hemophilics they have been mixed. When we mix the two serum of this category peoples, in such mixing there is a precipitation occurred between the serum, and this occurred because of the presence of this antigen, and that antigen firstly detected in Australian people. Because of that, this antigen is known as a Australian antigen. But later on in 1968, this Australian antigen, which was detected by Bloomberg, it was found to be a part of hepatitis B virus or serum hepatitis. So because of that, this name of Australian virus, it has been changed to a hepatitis B surface antigen, HBsAg. This is the short form of hepatitis B surface antigen, and this antigen is earlierly known as Australian antigen, but later on it is replaced by this hepatitis B surface antigen. You can see there, this is the hepatitis B virus, this is the core antigens. Now in this virus there is a presence of two envelopes are there, and these envelopes protect this virus from our immune system. This is the envelope, first envelope, outer envelope. This is the core envelope which protect the inner genetic material of the virus, and this envelope, it is made up of three different proteins: small surface protein, which is acting as antigen, medium surface protein, and the large surface protein. Along with this, there is a presence of a DNA polymerase enzyme which is present in the center core of this virus. And along with this surface antigen, there is a presence of another type of antigen. That antigen is known as E antigen. And this antigen, it is denoted as HBe antigen. And in the center, there is a presence of DNA molecule, which is acting as a genetic material of this hepatitis B virus. Now, when this virus, it is observed under the electron microscope, which is taken from the patient, three types of particles they have been observed. When this hepatitis B virus, which is collected from a patient, it is observed under the electron microscope, it was discovered that there are three types of particles are there. The most abundant form of particles, they are spherical, which is having size of 22 nanometer in diameter. The second type of particles, these particles are there, they are filamentous or tubular with a diameter of 22 nanometer and having variation in length. And these two particles, spherical and this filamentous particles, they are antigenically identical. And the surface components of the hepatitis B virus. And these are produced in great excess on the surface of this hepatitis B virus. And the third type of particle which is observed there, which are few in number and is a double-walled spherical structure having diameter 42 nanometer in diameter. And these three particles, first spherical, then the second filamentous, and the last one which is a spherical but having a double wall, this constitutes the complete hepatitis B virus. And this complete hepatitis B virus structure, it was firstly described by the scientist Den in 1970, and because of that it is also known as a Den particles. This third particle which is discovered, that third particle, it is also called as Den particles because of the discovery which is carried out by this scientist Den. So this is the morphology of this hepatitis B virus, which contains three types of particles. First one is a spherical having size 22 nanometer. Second one is a filamentous or tubular structures having diameter 22 nanometer but having variation in length. And the third particle, which is also spherical but having size larger than the earlier particle, that is 42 nanometer in size. And this particle, third particle, it is known as also by another name, that is Jane particles. Now these envelope proteins which are present on the surface of this virus and the surplus 22 nanometer diameter sphericals and filamentous particles is whole the structure. Envelope protein, then these spherical and filamentous particles, they form the hepatitis B surface antigens. And this presence of this hepatitis B surface antigen, it is very important. And this will we will see this what is its importance in laboratory diagnosis. Now this hepatitis B surface antigen consists of two major polypeptides, and one of the which is glycosylated. Means there is a presence of glycosyl group is there in one of the polypeptide which is forming this hepatitis B surface antigen. Now when we treat this virus with mild detergents, it disturbs the viral envelope and exposes the core of this virus or nucleocapsid of this virus. And the antigen which are expressed on the core, that is inner core of this virus particle, that is known as hepatitis B core antigen, which is present inside the surface envelope of this hepatitis B virus. A third antigen, which is known as hepatitis B E antigen, it is a soluble non-particulated nucleocapsid protein which is present there, and both this hepatitis B core antigen and hepatitis B E antigen, they are immunologically distinct. They are totally different from one another in structures and their characters. But these two antigens, they are coded by same gene. Still these two are different, but they are coded by the same gene of the virus DNA. Now this nucleocapsid, it encloses a viral genome which consists of two linear stranded DNA molecules which are held in circular configuration. You can see here in the structure, you can observe this. These are the linear molecules but which are held in circular configuration. And this DNA molecule which is observed in case of this hepatitis B virus, that shows two types of characters. One strand of this DNA, it is an incomplete strand, and that incomplete strand, it is called as a plus strand. And this DNA molecule, which is showing a one strand, in that DNA molecule there is a one strand is the incomplete one, and because of that, this DNA of this hepatitis B virus, it appears partially double stranded and partially single stranded. Means in this virus, the DNA molecule, it is partially double stranded is there and partially single stranded is there. And when we observe this, it is found out that this plus strand of this viral DNA contains a enzyme with them. That enzyme is known as a viral DNA polymerase enzyme, which is present along with the plus strand of that DNA molecule. And another characteristic is there, and this DNA polymerase enzyme, which is present there with the plus strand, it has two characteristics. First characteristic is that this enzyme is DNA dependent DNA polymerase enzyme. Means this DNA polymerase enzyme can participate in the replication process. And the secondly, that is RNA dependent reverse transcriptase function. So this single enzyme, it performs two kinds of function, reverse transcriptase function as well as polymerase function. So this is all about the etiological characteristic or morphological characteristic of this hepatitis B virus. Now its resistance characteristics. This hepatitis B virus, it is relatively heat stable. Means it cannot be easily destroyed by heat. It remains viable at room temperature for longer period. If we heat some objects up to 60 degrees Celsius temperature for 10 hours, and that object carries this hepatitis B virus, it reduces the infectivity of this virus by 100 to 1000 folds. Means that virus become inactive. It is not capable of causing infection to the healthy person. When we treat that object which carries them at 60 degrees Celsius temperature for 10 hours. Similarly, this virus is also susceptible to some of the chemical agents. When we expose this virus to hypochlorite or 2% of glutaraldehyde, it inactivates this virus infectivity. But the surface antigen of this virus, it may not be destroyed by such treatment. Means these surface antigens which are present on the surface of this virus, they remain as it is. They are not destroyed by the treatment with glutaraldehyde or hypochlorite. So this virus is capable of surviving heat for longer in room temperature. It can remain for longer time, but it can be inactivated by using 60 degrees Celsius temperature and 10 hours treatment. Similarly, this is also susceptible to chemical agents such as hypochlorite and glutaraldehyde. So we can easily inactivate this virus when it is present in our household conditions. Then the next point in this hepatitis B virus is the epidemiology. Now this hepatitis B virus, which occurs throughout the world, there is no seasonal distribution is there. Means in particular season this virus is there, or in next season it is not there. Such conditions, they are not there. Whenever it present constantly at that particular location. And this infection which is caused by this hepatitis B virus, it is a sporadic. It is not a continuous one. And though occasional outbreaks of this infection may occur in hospitals, orphanages, and institutions for mentally handicapped persons. So these are the most susceptible conditions, most susceptible host. Means in this condition, in this environment, this virus can cause occasional outbreaks. But mostly this infection, it is a sporadic, not continuous one. At some place it may there, and in different places it may not be there. So it is sporadic, but on some occasions it can cause outbreaks in hospitals, orphanages, and the institutions where mentally handicapped persons live. Now the prevalence of these hepatitis B carriers, they are playing very important role in epidemiology of this virus because they are the most important source of this infection. Because other than the human, this virus cannot affect the other animals. The only host for this virus is the human being. So this virus, it is maintained in the environment by the human carriers only. Now these human carriers, these hepatitis carriers, they varies widely in their distribution or their prevalence in different countries, and it relates to their living standards. We can see some of the conditions in overpopulated areas under the developed regions have endemicity more than where the carrier rate is more than 8%. And this is observed in some of the countries such as Africa, where the carrier rate is high. Where the carrier rate is very high. 8% of the carrier rate is there. Those persons which are infected with HBV virus, that is hepatitis B virus, in this Africa, South Asia, China, part of the South America, in these countries, these infected persons get converted into the carriers. There is a 80%, 8% is the rate of these carriers in these countries. Low endemicity means low rate. In developed countries such as Western Europe, North America and Australia, where the carrier rate of this virus is less than 2%. And the intermediate endemicity, where the carrier rate is 2 to 7%, which these countries include Eastern Europe, Middle East, South Asia and part of South America. So we can observe differences in the carrier percentage in different locations. In developed countries, the carrier rate is less than 2%. In some of the developed parts where the overpopulation is there, like China's, there is a higher rate of carrier development is there. 8% of the peoples which are infected with this hepatitis B virus may get converted into carrier state. But in intermediate countries where the carrier rate is in between 2 to 7%. And in this region, we are present in these intermediate regions. Now there is also difference in the occurrence of this infection to the chance. Rich and poor countries, they also differ in age and mode of infection. In rich countries, the infection mostly occurs in adolescents and young adults, through where these persons, these young adults and adolescents, they get addicted with the drugs. They can use the contaminated syringe and needles, through which this infection can be transferred in case of rich countries or developed countries. Again, there is a sex part is also there, through which this infection can be transferred in developed countries. While in case of poor nations or not developed countries, infections, they are usually occurring in the young ones. And this infection may acquired by that young one by perinatally from mother to baby, or horizontal among the children. The children which are playing in groups. In such conditions, that infected child may transfer his infection to the healthy child. Now this hepatitis B infection, it only occurs in human being. No other animal get infected by this virus, or no animal reservoir is there. The only reservoir for this infection is the human beings only. And this virus, it is maintained in the environment in large pool by carriers. And these carriers, they carry this virus in their blood, where it circulates for longer time, even life of that host. Whenever that host remains alive, up to that time, this virus remains in these carriers. So, these carriers be a good source of this infection for the healthy one. A carrier is a person. Now, how to define a carrier in case of this hepatitis B virus? A carrier is defined in this hepatitis B virus who is having detectable hepatitis B surface antigen in their blood for more than 6 months. Those carriers, those persons which are having hepatitis B surface antigen in their blood for more than 6 months, they are called as carriers of this hepatitis B. And following the infection of this virus, about 5 to 10% of the adults, and where 30% of the children and 90% of the neonates become the carriers. Now, you know that this infection, it mostly occurs in the young ones. And those young ones which are infected by this, that is 5 to 10% of these adults, they are infected. Then 30% of the children, they are infected. And 90% of newborn babies or neonates becomes the carrier. The carrier state is most common in case of males. Those males, they are most favorable to this, to become a carrier in this hepatitis B viral infection. Now it is estimated that 3,500 millions of the carriers are present in the world now. So this much amount of peoples, they have carriers, they become the carriers of this virus. And out of them, about 45 millions are in India, which has the largest second pool carrier, next only to the China. So you can see that large amount of carriers, majority of the carriers of this virus, they are present only in two countries. The first one is in China and the second one is in India. So these two countries, they have the majority of the carriers among the peoples which are living here in these countries. Now there are two types of carriers are there: highly infectious super carriers and the simple carriers. The super carriers are those carriers or those persons which are having high titer of hepatitis B serum B surface antigen along with hepatitis B E antigen, DNA polymerase and hepatitis B virus in their circulation. And in such patient, there is also elevation of a enzyme is there, transaminase enzyme. So super carriers are those carriers where there is a presence of high titer of hepatitis B surface antigen along with hepatitis E antigen, DNA polymerase and circulation of this hepatitis B virus. And those persons are called as carriers of this hepatitis B infection. Now this hepatitis B virus is a blood-borne virus because it is present in the blood system of the that carriers. And this infection can be transferred to healthy person by parental, sexual and perinatal mode. So these three modes, by using these three modes, this viral infection can be transferred from carrier to healthy person. Blood carriers, and often the less amount of the patients, those persons which are infected with hepatitis B virus and those which carry this virus in their blood, they are the most important source of this hepatitis B infection. It is also observed that this virus may be present in other body fluids, excretions. This virus can be detected in these excretions and in body fluid, such as saliva, breast milk, semen, vaginal secretions, urine, biles, and feces. In these fluids also this virus have been detected. And of this, the presence of virus in semen and saliva, they are known to transmit this infection. So this infection can be transferred by the saliva as well as the semen. And other modes, they are less efficient than the blood. It is also detected that through the fecal matter, this infection cannot be transferred to the healthy person. The only mode of the transfer of this infection is through the blood transfusion. So because of this characteristic, this hepatitis B virus infection, it is also known as a serum hepatitis, transfusion hepatitis. These are the other names which are used in relation to this hepatitis B virus. Now earlier, whenever the transfusion process has to be carried out, they carry out this transfusion process without testing for testing the blood of that donor. But nowadays, the transfusion of blood from carriers, from the carrier's blood to the healthy person, to the blood, it has been screened. Nowadays, various technologies are available through which we can screen the presence of this virus in that blood sample. If that blood is having this virus, we are not going to transfer this blood to that person which is needing that. So this has lessened the transfer of this virus through the blood. Now this infection, hepatitis B virus infection, it can be acquired by direct contact, where there is a presence of open skin lesions such as pyoderma, ischema, cuts, scratches. These are the various modes through which this virus can enter into the young children in developing countries. In household conditions, there is also transmission of infection, where there are opportunities exist there for contact with blood, saliva among the members. So this infection, it can be transferred through the direct contact with that patient, with that carrier. Now it has been known that this hepatitis B virus, it can survive in mosquitoes, bed bugs for near about 2 weeks after blood meal. After that blood is taken by that mosquito or bed bugs, it can remain in these animals, in these flies, for near about 2 weeks. But in this animal, this virus is not multiplied then in these mosquitoes and the bed bugs, and they don't appear to transmit the infection of this hepatitis B virus. So this virus, it can multiply in mosquito, in bed bugs, but it is not transferred to the healthy person. So these are not acting as a mode of transmissions. Similarly, congenital and vertical transmission is very quite common from carrier mothers. Those mothers which are having pregnancies, they can transfer this infection to her babies, and the risk of these babies, it depends upon the detection of the presence of this hepatitis B surface antigen. If the amount of this hepatitis B surface antigen it is high, then there is a high risk that her babies may have congenital type of hepatitis B infection. If the titer of this surface antigen it is low, then we can say that there is a less chance, 5 to 15% of the chance is that babies becomes infected with this hepatitis B viral infection. But that risk is very, very high if the titer of that surface antigen in that woman is in between 60 to 90%. There is a high risk that baby may have this viral infection after birth or before the birth. Okay, so this is all about the etiology, and